Okay, ladies and gentlemen, let's get started. We've got quite a bit to cover today. Just a couple of notes. We do have a test next Wednesday, and that seems really fast compared to how slowly the first exam came along. That exam's going to cover chapters 3, 4, and 5. I'll finish 3 today. I'll cover about half of 4 today, and then we'll finish up 4 and get into a little bit on skin. Chapter 5 is not very long at all. And those 3 chapters will be part of next week's exam. That exam's a full-length exam. It is going to cover the material on those 3 chapters plus vocabulary 17 through 36. I'm going to do about 6 of those vocabulary slides right now. I'll do the last 6 or so on Monday. Hopefully you've already made your vocab cards and you're not stressing about the vocab at all. On Mastering, you'll find 3 assignments, 1 for Chapter 3, 1 for 4, 1 for 5. 3 and 4 are out there. They're available to you. 5 will pop on in a day or so. No one's seen 5 yet. That'll be the last one that you'll do. And there will also be a quiz on Mastering due next Tuesday by midnight that will help you do well on the exam on Wednesday. Those who are listening, hybrid students, you got the email. Call the testing center for your testing time. Chapter 4. It's all histology. It is mostly review from what you have seen in the lab. Everyone now has been through the histology lab. Today starts the skeletal system lab, bones, and bones will be part of exam 3. Now, I'm going to say something right now. I'm noticing, and I'm hearing a lot from the adjunct lab instructors as well, that many students appear to be coming to lab unprepared. Meaning, yeah, you've got your pre-lab done, but you haven't listened to the video. And if you're not listening to the video, then you're not understanding everything that's going on in lab, and we're assuming that you've listened to that video, and I'm going to assume as I move into this presentation that you've done that. And if you haven't done that, the dominoes are going to start falling backwards on you, okay? So make sure that you are going to lab prepared, and that includes listening to the video that's posted in the lab folder. You should come in with your PowerPoints kind of filled in and with notes and questions. Your instructor is there to answer questions about the presentation, not to redo the presentation, okay? So make sure you're getting in that habit. It will bite you in a bad way if you're not coming in prepared week by week. Let me jump back to vocabulary. And I believe I need to pick up with you folks on 23. And I'll go through 30 today, and then the last 6 or so on Monday. So derm, skin. Dermatology, the study of skin. Detruse, you have around your bladder a muscle called the detrusor muscle. And when you empty your bladder, you're forcing the urine away. Dextro, to the right of. Di is 2. You've seen the word diploid, or you'll hear it again today. Di, 2 copies of the chromosomes. And dia means through or across. Think diameter, right? The meter, the measurement across something. Diastole. In the heart, we'll talk about diastolic pressure. That is the time when the heart is resting, or the stand-apart time. Dips is thirst. Polydipsy would be excessive thirst. Dis is apart or opposite or absent. So, for example, to dissect is to take apart, or disinfect is to be absence of infectious agents. There's that dipple again. DIPL means double. Diuret, you take diuretics to urinate. Dorse, you know, means back. Duct, as a prefix or suffix, means to lead or to draw. And when we get to the brain, we'll see there's a layer around the brain called the dura mater. It's kind of a hard, tough layer. So dur means hard. Dynia is pain. Dis, also painful, difficult or bad. So dysuria would be painful urination. E, as a prefix, out or from. And eel, pertaining to, much like ac, right? Eel and ac both mean pertaining to. EF, away from. We'll hear the word efferent quite a bit this term. Efferent, going away from something. Ecto, outside. You'll hear me say ectoderm today. Ectomy, to cut out. Appendectomy, to cut out the appendix. If you see edeme in a word, or EDE, it's some sort of swelling, some sort of edema. If a word ends in EL or ELLE, it means small. Organelles, you appreciate, are like small little organs inside the cell. EM and EN both mean in or into. Embol, stopper. If you have an embolism, it literally is a blood clot that goes to the lung that stops the blood. Emesis, vomiting. So hematoemesis would be vomiting of blood, right? Dysemesis would be painful vomiting, if you put those words together. Emia, some condition of the blood. Encephal, brain. Endo, within. We've seen the endocrine system secreting hormones within the body. Entero, the intestines. Enteritis would be inflammation of the intestines. Itis means inflammation. And then I'll finish up with this today. Epi, on or upon. The epidermis is on the dermis, on or upon. Erythro, like your red blood cells, red. Estha is sensation, and EU is good. If you give a eulogy, you're giving a good word. If you see the word eupnia, eupnia is referring to good or normal breathing. So that'll finish up, and we will go through Glyco, so 6 more slides, on Monday. So go ahead and get those done, and be looking through those cards every day. And by the time the test comes, you'll be okay. Now, the students who have struggled with vocabulary, again, when you're looking at the words I give you, find the parts that you've seen, right? Just kind of put a slash there and then write those words in on the blanks. It'll be very similar to the last test. It is not cumulative. It'll only be from these slides 17 through 36. Going back now to Chapter 3. And we had just gotten to the idea of meiosis. I mentioned apoptosis in the formation of the fingers and toes. And now over to tumors. That's where we dropped off, as I recall. Is there anything in that conversation up to that point? Chapter 3 was about organelles, structures of organelles, and I was just kind of giving you a couple aside topics. And I want to move into tumors. Anything at all? Yes, ma'am.
When you said that on the review sheet, it says like the question was like the structure of the chemical structure of the plasma membrane. Do you just mean like the
proteins and lipids?
Oh, okay.
Proteins and lipids. The question was what's the chemical structure of the membrane? So think there's phospholipids, right? And then there are proteins, integral and for peripheral, also found in the membrane. And there was some cholesterol, right? Those were the major issues, the major members of the membrane. So we've talked about, or we'll be talking about, the cell cycle more. And the cell cycle is important when you start thinking about tumors. So tumors are basically when cells go out of control, right? When cells no longer behave the rules, they no longer are dictated by the rules of the cell cycle. There's some imbalance. And you get a tumor or a neoplasm, a solid mass of cells. Now, not all cancers make a solid ball of cells. Leukemia, some blood cancers just have an overabundance of a cell type. But here I'm talking about a solid ball that would form a tumor of some sort. And cancer tumors come in basically 2 types. This is very simplified, but there are benign neoplasms. Now, if you get a benign tumor, this is good news, right? Benny means good, and literally, this is pretty good news. It typically will not kill you unless this tumor is deep in the brain and they can't get to it, or perhaps it's pushing up against a vital organ, and again, they cannot surgically remove it. But typically, a benign tumor is good. The characteristics here are this. The benign tumor stays confined within a connective tissue capsule. So this tumor kind of stays within a space, and it doesn't spread from there. So typically, if possible, they can surgically remove this tumor, remove the entire thing, and all is well. You need to go back. One of the things that tumors do is that these cells become de-differentiated. Now, when you're an embryo and your cells become liver and muscle and kidney and brain cells, that term used to describe how the cells become different is differentiation. The cells are differentiating. They're becoming different from one another. In a tumor, the cells, they go backwards. They become less specialized. They become more adolescent or more childlike, and they start growing faster as a result of that. So they de-differentiate. They become less specialized. They also have an uncanny ability to increase their own blood supply. If you've got a solid ball of cells, that solid ball of cells can't survive unless there is blood supply and nutrients going to that. So cancers have a way of increasing their own blood supply. Now, the other group of cancers are malignant neoplasms, and these are the ones you don't want to hear about. These are unencapsulated, meaning they don't have a secure edge, and they tend to travel and break off. They also de-differentiate. They also increase their vascular supply. They also grow rapidly, more rapidly than most benign tumors. And here's the kicker, they tend to spread. Because they don't have that encapsulation, they tend to spread, and that term for spreading is metastasis. They metastasize, and they spread to other places in the body. They spread there via blood, or they spread there via the lymphatic system. So a tumor that starts in the lung may spread to the brain, or a tumor that's in the bone may spread somewhere else.
So it's if it's in the lung, it's like the cells keep multiplying until they get to the brain, or is there something in the cell that's traveling up there?
So the lung tumor would develop until some of those cells broke off and traveled to the brain.
Oh.
Yep. So again, cancer, you're losing control of the cell cycle. I've already said these words. Oh, the other thing is cancer cells lose their contact inhibition. What that basically means is this. If I took a cell out of my body, out of your body, and I put it into a petri dish, and that cell started to divide, when that cell reaches another cell and touches another cell, normal cells will stop growing. They know that when they touch each other, that's sort of like the rules. We stop growing at that point, I've got my space. However, cancer cells don't have that inhibition, and they begin to grow on top of each other, making that mass. So there are a lot of things, right, that are going wrong in a cancer cell. So again, they differentiate, and the word for increasing their vascular supply is angiogenesis. You can break that word down. Angio, means vessels, and genesis, in the beginning, the formation of. So angiogenesis is the formation of new blood vessels. Again, they travel, they invade, they go to places where cells normally would not travel. And that term is metastasize or metastasis. So that's just a little bit about tumors. So I hope you appreciate why the cell cycle is so important. It's not just this academic thing about every cell, but when the cell cycle is disrupted and cells are out of control, we've got some major issues on our hands like tumors. Now, a couple of the details on this are not to be worried about. This is not for a quiz. This is for not for next Thursday. This is for exam number 2 next Wednesday. And in my email to you, you saw me mention that there's a short written part on this exam. And this is that written part. It's basically you're going to write 3 sentences. I'm giving you 11 terms here. And of these 11 terms, you're going to use 10 of them. And you're going to write 3 sentences correctly linking these terms together. Some of these terms you've not yet heard. They'll be part of our conversation today. Others you've already heard of. You already know, for example, nucleus, DNA, cytoplasm, ribosome, protein. Those you've heard. The other terms will be coming up today. So your job will be simply to write sentences using these words, linking them together. It's basically like you're reading a textbook, and at the end of the chapter, there's a little review. And in that little review, they have very nice sentences with lots of words that are usually highlighted or bolded about ideas that have been in that chapter. So, for example, proteins are produced by ribosomes that are found on the rough ER. Right? That'd be a sentence. I just used 3 of the terms. So it's not hard, I don't think. And if you can link these words into sentences, then I know you understand their relatedness. I will give you the same 11 words on the test. You don't have to memorize those. I'll give you a little blank space on the test, and you'll write your 3 sentences. I usually get the question, what is a sentence? Well, it starts with a capital letter and ends with a period. Okay. What is not allowed is, in Dr. Macaulay's class, we learned about transcription, the nucleus, DNA, and translation, period. That will not be allowed, right? The sentences must talk to each other. They must communicate. The 3 sentences do not need to be linked. In other words, it doesn't have to create a paragraph that flows, but simply 3 statements that link these words together. As you're writing the sentences, underline them. The only reason you will miss points on this is if you don't underline words, because I'm not going to go looking for your 10 words. Okay? I'm just not going to have the time to go looking for them. So if they're underlined, I'll count them, and then I will make sure that what you wrote makes sense. Full credit. Question?
What if we have more than 3 sentences?
No more than 3. Because what I don't want is 10 definitions. Right? I don't want a definition of transcription followed by a definition of nucleus followed by a definition of DNA. So 3 sentences is your limit. You can do 2, but no more than 3. Okay? So you're forced to put the words in together into a sentence. Okay? Now, the other words, again, the word that doesn't work well for you, throw it out. Just 10. If you use all 11, that's fine. I'm not going to give you any bonus points, but you get at least 10 words, okay? So I'll mention that again to you on Monday. You can practice it. You can write it out now. You can have somebody else read it. And then on the test, you may not bring that in. but you may just simply rewrite from your memory what you wrote. Fair enough? Understand that little part? It's worth 10 points, and I hate it when I see people not do well on that. They just didn't prepare. So just look through that, and you'll be fine. Okay, moving ahead. So the first part of Chapter 3 was about the organelles. Now the focus is going to be on DNA. We're going to turn our focus exclusively on DNA, and we're going to see, you know that DNA is wrapped up in the cell as chromatin. Chromatin is just a fancy word for the DNA wrapped around the proteins. And then you know that that chromatin becomes visible under the microscope during mitosis, and that's the only time where you can see these chromosomes. Now, no biology class is complete without mentioning Watson and Crick. These two guys, about 60 years ago or so now, came up with the structure of DNA. It was big news. It was a big deal. It's still recognized as a big deal. Before that, we knew about DNA, and we knew about proteins, but no one really understood what was more important, DNA or proteins. Since that 50, 60 year ago discovery, it's amazing what has happened in science and in medicine revolving around DNA, to the point now where we can predict diseases and we know gene structures, and it's just really, really amazing. We even now have medicines, cancer treatments based specifically upon the DNA structure of the mutation related to the cancer. So a lot of things have happened over the last few years. Now, they did not do any real experiment. They actually just built a model. Now, they were genius in doing so, and they actually ripped off ideas from other scientists. So they didn't do this in a bubble by themselves. They actually used data from this young lady, Rosalind Franklin. And what she did is, before Watson and Crick could do their model, she took DNA and shined X-rays on it. Now, I'm just oversimplifying this, but when you do that, you get this pattern, sort of like an X-ray, if you will, of DNA. And what this pattern told her is that DNA is of a set diameter, right? It's a certain size from side to side, and that it's a helix. Before that, we didn't know that DNA was a helix or that it was a fixed diameter. So Watson and Crick used some of that data to figure out the structure of DNA. The Nobel Prize was awarded to Watson and Crick in 1962, but she had already passed away, probably from shining too many X-rays in her face. And so she died early, and they don't give the Nobel Prize to those who have already passed away, so she did not get the award, unfortunately. You know a little bit about DNA versus RNA. So let's recap that. You know that DNA is typically double-stranded, RNA is single-stranded. That we got from Chapter 2. You know that RNA has A, C, G, and U, whereas DNA has A, C, G, and T. And there's a different sugar group. DNA has deoxyribose, whereas RNA has ribose. That's where the name comes from, right? Deoxyribonucleic acid or ribonucleic acid. And you're not going to have to recognize the structures here. Just so they're very, very similar. They're very, very similar indeed. So here's that double-stranded structure of DNA. Again, sugar phosphate on the backbone. And the nucleotide bases are sticking out. A's binding with T's, C's and G's coming together. The dotted lines represent the hydrogen bonds that are helping to hold them together. So C's and G's are held together by 3 hydrogen bonds, A's and T's by 2. And what you also see here is that a T always matches with A, and G always with C. That was part of that magic that she discovered, that DNA is of a set diameter, right? You could not have 2 A's matching up together because the molecule would be too broad. And you couldn't have 2 T's matching up together because the molecule would be too skinny. And so part of that magic in their making of the molecule structure of DNA was from this connection. These bases belong to 2 different families. Some are called purines, some are called pyrimidines. A and G, the big blue guys here, are the double molecule. They are the purines. The pyrimidines are a single circle structure, the green ones. The way to remember this is pretty simple. The pyrimidines looks like the word pyramid, and to make the pyramid, somebody had to cut the stones. So C-U-T, those 3 are the pyrimidines, and the other 2 are the purines. And what you see is that, again, one purine matches up with one pyrimidine along the length of the DNA molecule. So I want us to think about DNA for the next few minutes. DNA is found in the nucleus. In your cells and mine, we have 23 pairs of chromosomes. That is 46 chromosomes altogether. And remember that every cell goes through the cell cycle and will eventually divide to make new cells. Somewhere along the way, there was the S phase. And the S phase is the time that stands for synthesis. And it's during this time that the RNA is copied. Now, the more appropriate word for that is replicated. The DNA undergoes replication, or is replicated. Remember that when a cell goes into mitosis, it has already copied everything. And all it's going to do now is just simply split the pie in half. Right? It's going to take those chromosomes and take the copied chromosomes and put them into each of the new daughter cells. It's going to take all the organelles and also separate them so that each of the new daughter cells starts off with a full complement of DNA and organelles. How does a cell do this? It's pretty amazing. What Watson and Crick discovered, what they hypothesized, I should say, is that A's always go with T's and C's always go with G's. And that's exactly what happens, is that when the DNA is being copied, it's that simple. The cell looks and says, okay, this is a C, so I'm going to make a copy of a G. So it just kind of does this complementation, this pairing up. So A with T, C with G. And you're going to see that over and over and over as we go through this. Now I'm simplifying this, and I'm going through this rather quickly. Because I'm not worried about the specific details of this. Most of you have had what I'm going to talk about somewhere along in a biology class. You've forgotten some of the details. You can take an entire semester's course learning about replication and transcription and translation. So I'm taking a very vast amount of material and squeezing it down into just an overview. Replication, the copying of your DNA. This is an amazing story. In our cells, the fastest that a human cell can divide is about every 8 hours. Now, in order for a cell to divide, it must copy all 6 billion ACTs and Gs. Right? There are 6 billion ACTs and Gs in your cells. And every one of those must be duplicated or replicated in order for a cell to copy. So it's a pretty amazing process. In us, that process occurs at about 50 nucleotides per second. So 50 ACTs and Gs per second are copied, and that copying better be good, right? It better make a really good copy, because if mistakes are made, those are mutations, we won't live very long. So it's important that the copying is very, very accurate. Now, bacteria do this even faster. They can do about 500 nucleotides per second, and in fact, almost everything we know about replication came first from understanding bacteria. So bacteria have been very, very important in understanding this. But we're only worrying about humans. So what happens if replication goes wrong? What happens if the copying of the DNA is not as perfect as it should be? Well, then we end up with a mutation. A mutation is basically an error in the copying of the DNA. And replication makes a mistake about every 100,000 letters. That's pretty good. I mean, if Toyota only had one lemon out of every 100,000 cars, that would be a phenomenal record of success. So great quality control. But when you consider that we have 6 billion letters to copy in every cell, making a mistake every 100,000 still leaves quite a few changes to have occurred. So a lot of mutations do occur in our cells naturally as our DNA is copied. Now, if that mutation happens in a somatic cell, and a somatic cell is just a fancy word for one of your body cells. Soma means body. So all of the cells of your body. If you get a mutation in your skin cell, big deal. You're not going to give it to your kid, right? If you develop skin cancer, you're not going to pass that skin cancer on to your children. So any changes that happen to your somatic cells are not going to be heritable. However, if that mutation occurs to your germline, that is the cells that will make your egg or sperm, then that change is heritable and can be passed on to your children. To show this idea of a mutation, on the left is a typical, just a cartoon of a double-stranded DNA molecule, and you see here A's and T's matched up and C's and G's matched up, and somewhere in this copying process, a single change is made so that a CG pair becomes an AT pair. That's the kind of mutation that I'm referring to. It's a change in the DNA sequence. Now, these changes can come in two basic types. I mentioned chromosomal mutations last time. That is, for example, errors during meiosis. And this is where we get the little child with Down syndrome, right? Something went wrong with the chromosomes overall combining. during meiosis. I don't want to talk more about meiosis right now. I want to talk about the bottom, about point mutations. And these errors happen through replication errors, right? The machinery just doesn't get it right all the time. These changes also can occur from your environment. So in our environment right now, we are being bombarded by chemicals, by radiation, by X-rays, by ultraviolet radiation and UV light. Going to the tanning booth, right? Inhaling smoke, breathing even, has particulates in it, all of which can cause mutations to our DNA. The good news is, even though we're being bombarded by things that can cause mutations, our body has a lot of repair mechanisms that can fix most of those errors. So as mutations are occurring every second in our body can fix most of them. It's like an old-fashioned typewriter. Basically, you're typing along, oops, I made a mistake, backspace, fix it, and go on. And that's basically what our cells are able to do for most of the mutations. But some mutations do get maintained, right? They stay with us. Each of us, on average, carries 5 to 10 mutations that can cause disease. Now, we may not ever see that disease occur in our body, but if we have a child with somebody else who has that same mutation or a similar mutation, then our children may be at higher risk for those particular diseases. Now, this number is probably low. In fact, I know it's low. Now, we are beginning to understand that there are genetic conditions or genetic connections to heart disease, genetic connections to liver disease, genetic connections to diabetes. That was not understood 25 years ago. So now I would argue probably all of us carry probably 10, 20, 30, 50 genes that could possibly cause disease as we begin to understand the influence of genes on our health. Where did those mutations come from? Either we inherited them from our parents, right? They already had the mutation and now we've got it, or a newly arrived mutation that occurred in our body. That is kind of demonstrated here. As this graph is showing the different types of cancer in men over time, and you can see that by age 80, prostate cancer is a pretty significant type of cancer in men. Skin, rectal, esophageal, lung and cancer is not even shown on this. But what you can clearly see from this diagram is that with age, cancer rates go up. Why? As we get older, we have accumulated more mutations in our own DNA. Right? And so if I go outside and I get too much sun and I get an epithelial cell in my right wrist, that's my left wrist area, right? And I get a mutation in that cell. Now that cell has a mutation. Maybe nothing happens, but now that skin cell is going to divide, right, and make 2. So now both of those cells have that mutation. And then those 2 cells are going to divide and make 4. And those 4 cells now all carry that mutation. Now, if that same cell that already has one mutation now gets another mutation, and I start accumulating mutations. And we know that it's the accumulation of these mutations that can lead to cancer. Okay? Now, some people are born with some of those genes already. So if you have a family history of cancer, what it means is that your family tree is already giving you a gene or 2 that makes you higher predisposed to cancer. Other people are born with a clean slate with some kinds of cancer, and they have to accumulate these mutations over life. Others, like when you have a childhood cancer or a cancer that comes early in life, it suggests 2 things. One, they were born, they inherited some of that mutation, but then there was something, chance, habits, whatever, that contributed to other mutations that occurred. And so they got cancer younger in life. 25 million cells every second are undergoing mitosis in your body. 25 million, right, cells are undergoing mitosis. That means that 25 million cells are dividing. That it suggests, too, that 25 million cells are undergoing apoptosis, right? They're dying because you'd have too many cells if they kept growing. Now, when a mutation occurs, right, I told you, 25 million cells every second, and every 1 out of 100,000, there's an error. So there are a lot of mutations occurring right now in your body. There's 3 choices. 1, your body can repair that damage. I told you your body has about 130 different ways of repairing these mutations. So number 1, the mutation occurs, the body fixes it, life goes on, no problem. Number 2, the mutation can occur, the cell can't or doesn't fix it, and the damage is so substantial that the cell can no longer survive. So the cell undergoes apoptosis, right? It kind of does cellular suicide and kills itself. So now that's taken care of the mutation as well, hasn't it? The third thing is that you're just stuck with it. That change in the DNA becomes a permanent a change in your genome, in your DNA. So I think of this as sort of like my family, right? I can either fix them, kill them, or live with them. Right? Those are the three choices that we have, right? And that's the same three choices that your cell has to deal with mutations, right? Fix it, kill them, or live with it. Now, these changes over time are what is the basis for Darwinian evolution. So the changes that occur over time are thought to be an accumulation of these gradual changes from mutations. I want to get to a punchline here at the end of this part of Chapter 3. And to do that, I need to describe to you two processes. That is transcription and translation. That is, how in the world is it that you go from DNA found in the nucleus, and how is it from the instructions in the DNA one makes protein? Now, again, this is a very complicated story, and I'm going to tell you just enough information so that you can retell the story to your grandkids in a very simplified way. Okay, big ideas. So DNA undergoes a process called transcription. Transcription is taking the DNA and making an mRNA copy. To transcribe something is to copy it. Some of you, if you copy your notes, right, from lab or lecture or from another class, another way of saying that is that you transcribe your notes. You basically copy them again. So transcription is the copying. And then that mRNA, that copy that you're making, is going to be translated into a very different molecule that is proteins. Now tell me, where must transcription occur? Where in the cell must transcription occur? You're starting with DNA, right? And you're going to RNA. And DNA is only found in the nucleus. So that's where this must occur. This process of transcription occurs in the nucleus. Now, here's where the real cool mystery is. I think this is cool. Every one of your cells, every one of your trillions of cells has in its nucleus, if I could unravel all of your 46 chromosomes, your DNA would be about 6 feet long. Right? So you'd have about a 6 feet long, thin thread of nucleotides, A, C, T's, and G's. Of that 6 feet, only about 1 inch actually codes for proteins. The other 98, 99% of your DNA is said to be junk or non-coding or stuff that we don't think is that important. Now we're learning that a lot of that, quote, junk has very important processes, right? Very important things are happening. But for the most part, we still know that only about 1.5 or 2% of your DNA actually is responsible for making proteins. Just to give you a visual of this, your DNA has parts that matter and parts that don't matter. That's how I'm going to describe it. Parts that do make proteins and parts that don't make proteins. The parts that do make proteins are called the exons. The parts that do not make proteins are called introns. Now, I apologize for whomever made up these terms. I think they're bass-ackwards. If I was going to be naming this, I would call the exons, I need to go back. The exons are the part that do code for the protein. I would not call them exon. Exon sounds like it's going to exit and not be important. The stuff that's junk here is called introns. Well, intron sounds like something that would stay in. Nope, that's what leaves. So the terms are kind of backwards in my mind, but you just need to learn those. So the introns are the junk, the exons are the good stuff, and what happens is that the DNA, this long 6-foot molecule, can sort of piece together the good stuff, get rid of the junk, and make the important protein sequences. So here these intron purple structures are looped out and lost. The exons are linked together to make the mRNA. Now, there's 2 more terms I need to describe to you that are part of those 11 words. One word is genome. That's not one of your terms, but it's going to make sense to you in a moment. The genome. The genome is all of the cells that are in your, all the genes that are in your cells. So that's the genome. And we have about 20,000 genes. You've heard of the Human Genome Project. So there's about 20,000 genes in you and me. All those 20,000 genes make about 100,000 proteins. All of those proteins are referred to as the proteome. Proteome is one of your 11 terms. So proteome is simply all of the proteins found in your cells, in a human cell. That's the human proteome. Now, 25, 30 years ago, everyone was convinced, well, because there's 100,000 because there's 100,000 proteins, that there must also be 100,000 genes. It was thought that every protein was made by its own gene in your DNA. Now we know that's not the case. We know that one gene can actually make multiple proteins. And here's how it works. Again, the gene, that sequence of your DNA, has parts that code for protein, the exons, and pieces that don't code for protein, the introns. And the cell has the ability of connecting different sections together. And so here, you can see that sections 1, 2, and 4 have been connected to make one protein, while down here, different sections 1, 3, and 5 have been connected to make a very different protein. All this means is that we don't have as many genes as we thought we have. We only have about 20,000 genes, and those 20,000 genes can be recombined to make about 100,000 different proteins in your body. So that means that the genome is smaller than the proteome, right? There are fewer genes than there are proteins in the body. So let's make sure we understand this transcription. It is, again, the process of taking DNA and making from it mRNA. Now, you've heard me say RNA. The kind of RNA that is made here in transcription is called messenger RNA because it's going to carry the message to make the protein. This has to happen in the nucleus, as I've said, because DNA is found in the nucleus. This is a simplified picture of this. We're looking inside the nucleus. So the nucleus is here. This is actually a little bit of the nuclear pore on the outside. So this would be the cytoplasm out here. And this is the nucleus. And in the nucleus, we have some DNA. The DNA here is a double-stranded molecule. And you can see that the double-stranded molecule kind of bubbles open, and from it, a single-stranded mRNA molecule is made that's shown in yellow. Recall, I've told you that typically DNA is double-stranded, mRNA is single-stranded, and you're seeing that single-stranded molecule here. So that is transcription. We've taken DNA, and from it we've made a single-stranded mRNA molecule. The next thing that's going to happen is that we're going to take this mRNA molecule and we're going to use the instructions on it to make protein. Now, again, this is what Watson and Crick so famously described in their paper. They said, if this is the structure of DNA, then it must be that DNA is used to make mRNA and mRNA is used to make protein. And they were right. And we've learned a lot more details about it since then. But this sequence of how this whole thing works was hypothesized with rather uncanny accuracy in their paper. Here is the whole story. Here's the whole story shown in a picture. So again, we start off in the nucleus. We've got our double-stranded DNA. It makes the mRNA, and that is the process of transcription. Remember that the nucleus has those nuclear pores, and there's those big gaps in the nuclear membrane. And through one of those nuclear pores, the mRNA squiggles through the opening. Right? And now where is the mRNA? The mRNA is out in the cytoplasm. Now what do we normally think of as being right outside the nucleus? What organelle is usually right outside the nucleus? The rough endoplasmic reticulum. Now that's not shown here. Okay? But typically there'd be rough ER right outside the nucleus. Now why is rough ER rough? Because it has ribosomes. So we've not shown the ER, but we did show the ribosome. That's this purple dude. So this is the ribosome. It's sitting right outside the nucleus. And the mRNA is now associated with that ribosome. That ribosome is now going to go through the process of translation. It's going to take the message on that mRNA, and it's going to translate it and now create from it a specific protein molecule. That protein molecule is shown in green, and to make proteins, you must have amino acids, which means that out here in the cytoplasm, you better have a collection of amino acids so that you can build those proteins. So you've got transcription followed by translation. These two terms are in alphabetical order. So transcription does come alphabetically before translation. You just got to go out about 5 letters to see the alphabetizing of these. So if you're getting confused, right, transcription does come alphabetically before translation. Here's another picture of this. It's the same thing. And just by chance, the authors used the same colors, and I like that so you won't be confused. So here in the nucleus, I see my DNA. This just is a small representation of your chromosomes. From it, we make mRNA. This number 1 is what process? Transcription, right? So number 1 represents transcription. And then that mRNA is going to squiggle its way through the nuclear pore, find itself out in the cytoplasm, interact with the ribosome, and that process of making protein, number 3, is called translation. Okay. Number 2, no name for that for us. Okay. So this number 1 is transcription. Number 3 is translation. Now, having shown you this, I go back to our sentence instructions. And now when we go back to the sentence instructions, you see now that I have now included all these words. You now have transcription and translation. I've introduced the word proteome to you, and you already knew ribosomes and proteins in rough ER. I've just mentioned mRNA. Genetic code, I'll get to in a moment. I kind of cheated. I did not really say genetic code yet, but I will in a moment. And cytoplasm is definitely done, right? So we've got all the terms now. Okay? So you've got the terms. So now you can start thinking about how to put those sentences together. Another example would be transcription occurs in the nucleus where DNA is transcribed into mRNA. I mean, there's a sentence with 4 terms, right? So it's not like it's a difficult assignment, but make sure you've got that down for next Wednesday. Okay, let me go back to our little story. Let me fill in just a few more details about this. More specifically, let's make sure we appreciate translation. Translation, again, is the second part of this process. It is going to be the synthesis of proteins from an mRNA. This is going to happen out in the cytoplasm where the ribosomes are found, and it's going to occur because the mRNA traveled out of the nucleus through the nuclear pores. The mRNA is going to bind to the ribosomes and is going to create the proteins. Now, there's a player in here that I haven't mentioned yet that I'll introduce right now, and that is tRNA, or transfer RNA. This transfer RNA is going to transfer the correct amino acid to the ribosome based upon the message sequence. So I'll show that to you in a moment. These words get confused. I know that transcription and translation all start sounding alike, and a lot of students struggle with these two terms. So let me help you maybe in a different way. Do you agree that DNA is one of the nucleic acids? Right? It's a nucleic acid. And as such, DNA is made up of really 4 things, A, C, T's, and G's. Right? And those are the 4 nucleotides. So basically, it's a 4-letter alphabet, isn't it? A, C, T's, and G's, somehow making this long 4-letter alphabet that we call our genome, our DNA. Now, what happens during the process of transcription? Transcription is right here. Then you make mRNA. Now, mRNA is also nucleic acid, right? It's still RNA, and it's still based upon a 4-nucleotide sequence, or 4 nucleotide letters, except here you have T's in DNA, and in RNA you have U's. But it's still the same alphabet, right? We made one change, but it's the same alphabet. It's the same language. It's still the language of nucleic acids and nucleotides. But then what happens? When this message is translated, it is going to go to a vastly different language. It's like going from English to Russian. We're going to completely change the alphabet, if you will. And now we're going from an alphabet of 4 letters to an alphabet of 20 letters. There are 20 different amino acids that can build your proteins. And that's translation, right? That's a word we know. We know translation. We're changing languages. right? From one with a four alphabet system to a 20 alphabet system as a way of thinking about it. Transcription, I told you, is basically copying, right? We're copying within the same language. So let's take a look. Let's look at this in a different cartoon. Again, if I tell you enough times, you'll believe me. So we've got here the nucleus. The nucleolus is shown in this picture for us. There's that DNA molecule, and it's going to undergo transcription to make the mRNA. That happens in the nucleus. The mRNA is going to make its way out the nuclear pore and now interact with a ribosome. In this picture, the ribosome is this big purple dude. This strand right here, that represents the mRNA. And what you see in a colored way is that there are 4 different colors on this mRNA. You see yellow, orange, green, and blue. Those yellow, green, orange, and blue represent what? A, C, G, and U. This is RNA. So that's the RNA. Then what you're seeing is this funky-looking molecule called the tRNA. It looks like a little lunar lander molecule, some sort of little space creature, right? And what you're seeing is all these circles. These balls are the amino acids, and they're floating out in the cytoplasm. And this is where the translation occurs. Look at this connection right here. There are 3 letters on the mRNA. Okay, so I don't know what the letters are here, but there's orange, blue, and green. That orange, blue, and green is going to match up perfectly with the tRNA that has the complementary color. So in other words, C goes to G and A goes to U. That specificity is going to dictate that this particular amino acid is brought in. In this case, it's a red amino acid. In the tRNA before it, this blue, green, green dictates that a blue amino acid is brought in. And I'm simplifying it without any names, just for you to see how this system works.
So are you saying?
Go ahead.
I mean, you said the blue, green, green makes the blue come. So are you saying like
Yeah.
the first color?
I'm telling you that these 3 letters,
Okay.
blue, green, green, and it could be like ACC,
Okay.
will always bring in this particular amino acid, which in this case is colored blue.
Oh. Okay.
Question?
Where does the tRNA come from?
Yeah, the tRNA is also in our cells. It is made, it's an RNA molecule, okay? It's also made of ACGs and Us, so it's also made by the cell. Just know they're there, okay? We're not going to go into where they're coming from. They're just being made by the cell like everything else that you need.
So are we bringing in the same amino acid?
Yep, so keep watching this. So what we have here is on the mRNA. The mRNA is based upon a grouping of 3. And that group of 3 letters, sometimes you'll hear the word triplet. More oftentimes you'll hear codon. It carries the code, the triplet code, or the codon. So every 3 letters on the mRNA represents a codon, and I showed you how it specifically binds to the tRNA. Those 3 letters, or those 3 nucleotides to which it binds, are referred to as the anticodon. So the tRNA has the anticodon, the mRNA has the codon. They match up. And this is where the specificity of this whole process comes into play. Let me show you how this works. So the top represents in red DNA. So this is our DNA sequence. Now what's missing here is that they don't have the double-stranded DNA molecule, but that red represents our DNA. I know it's DNA because it tells me, but I also see T's in it, right? So if you see any T's in it, you know that you're dealing with a DNA sequence. Now, when that DNA undergoes transcription to make mRNA, it does the same pairing up that we've seen. G will match up with C, and C with G, and A with U. So we get that kind of pairing, that complementation going on. And then, when that mRNA goes to the ribosome, it's going to be translated, and each of these 3-letter codons is going to dictate which amino acid will be built next into the protein. So here, CGU, this CGU will always have this protein, or this amino acid, arginine added. Now, you're not responsible for knowing the names of the amino acids. I'll give you a little table and show you how to do this little activity. and you'll understand where I'm going with this as we finish up this conversation. So, as a reminder, we talked about this in Chapter 2. Proteins are built of amino acids. Right? There are 20 different amino acids that link together to make your proteins. Each of these little rectangles, each of these little green rectangles represents another amino acid. Now, amino acids do not just stay as a long, slender, molecule, but these amino acids fold over and create a three-dimensional protein. Remember, I discussed how every protein has a specific shape, and without the shape, the protein can no longer do its job. So every protein is made up of a specific sequence of amino acids, which then allows that protein to specifically bind up and wind up and have a specific three-dimensional structure in the end. To get there, we've got to have three different kinds of RNA. So let me just clarify these with you. mRNA. That's the molecule I'm talking most about. That's the yellow single-stranded RNA molecule that you're seeing in the transcription stories that is made in the nucleus and goes to the cytoplasm to interact with the ribosome. The ribosomal RNA, this one's new for you. The ribosomal RNA is made in part by the nucleolus, and the ribosomal RNA is the building block, part of the building blocks of the ribosome. So that makes sense. The ribosomal RNA is a building block for ribosomes. And finally, tRNA, that's that lunar landing looking molecule that transfers or carries the amino acid to the ribosome and has the anticodon associated with it. Now, these are all mRNA. They're all made up of A, C, Gs, and Us. Right? They are just different types of RNA molecules in the cell. What's really interesting to me, it's another chicken and the egg kind of story. In order to make ribosomes, you need RNA. In order to make RNA, you need ribosomes and the rest of this equipment, right? So it's a really interesting chicken and egg situation for cell biologists. So what do we have here? I've already mentioned this. Nucleic acids, mRNA, become protein. We change the language. We translate it into a completely different language. Now, where does this translation occur? With the ribosomes in the cytoplasm. Has to be. Now, I showed you that the codon is a group of three, sometimes called a triplet. And another one of the geniuses of Watson and Crick is that when they wrote their paper about the structure of DNA, they also hypothesized that the genetic code going from DNA to protein must be involving a triplet code. They said that 2 would be not enough and 4 would be too much. Don't worry about the numbers or the statistics or the mathematics here. Just know that the genetic code is based upon groups of 3. And here's the table that I will give you that you'll need to be able to use. This shows you the 64 possible letter combinations. If each codon is 3 letters, right, A, C, G, or U, and there's 4 different possibilities, 4 to the third power is 64. So this is where your 64 possibilities are coming from. And what this says is that every time the mRNA has, for example, in it CAC, the ribosome will put in histidine, this particular amino acid. And every time the RNA has in it CCC, it'll put in this amino acid proline. So that's how you're going to use this table. And again, I'll provide this table to you, and I'll give you some more practice here in a moment. 3 of these codons are stop codons. They're shown here in white. That means that when the ribosome gets to this particular letter, UAA or UAG, that the ribosome knows to stop making any or adding any more amino acids. The protein's synthesis is done. So here's how this works. The DNA sequence was GCA, starting over here on the top left-hand corner. So the DNA is GCA. From that, the mRNA would be GCU. Do you see that? Do you see where that came from? Because this is what you're going to have to do on the test next Wednesday. I will give you a sequence of DNA. From that, you will give me the RNA sequence. Okay? From that, you'll then go to the genetic code table and tell me which amino acids that codes for. So, if you have CGU, when you look on the chart, you'll see that that's always going to be ARG, arginine. And when you see UCA, that's always going to code for this particular amino acid called serine. So that's what you'll need to be able to do on Wednesday. And I'll give you some more practice with this here in a moment. The reason this works is the genetic code is redundant but not ambiguous. What does redundant mean to you? Yeah, I mean, if I'm being redundant, and sometimes I am very redundant, saying the same thing over and over in a slightly different way so that you get the idea. So redundancy means that there's more than one way to say something. But the genetic code is not ambiguous. In other words, when I say something, hopefully I'm not being ambiguous. What I'm saying is there's only one way, right? I'm not suggesting that there's wiggle room in the interpretation. So let me show you what that means as far as redundancy and ambiguity. If you look at this genetic code, it's the same table, just flipped with different colors. It's the same information. If I look on the table here, I see that there's at least two different ways to say serine. In other words, the RNA can have AGU or AGC. There's two different ways to say the same thing, therefore the genetic code is redundant. However, every time the cell sees ACG or AGU, it will always put in serine. There's no ambiguity, right? There is redundancy. There's more than one way to say it. But every time you say a particular three-letter code, you will only get one answer. So there's the non-ambiguity portion of the genetic code. So let's look at the bottom. What amino acid would UUG code for? So on the bottom, UUG, let's find UUG on here. UUG, boom, 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 would give you this particular amino acid Lu. Okay? And what about CGG? CGG, yeah, here we go over here. Arginine. That's what you need to be able to do, okay, on Wednesday. It is quite simple, I think, but let's figure out why this is cool. Yes?
For Wednesday, is it just like the three letters, or will we have to know like arginine?
Oh no, I will give you the code, and it will all be the three letters, and you don't even have to know what arginine is. Okay? So I'll give you the same code that you just saw. Now the other thing about the genetic code is that it's nearly universal. What that means is that almost every single organism on the earth, bacterium, fungus, plant, bacterium, you, me, right? Almost every organism except for a couple flatworms in Africa use the same genetic code. What's cool about this is that means I can take a firefly. You know, fireflies have little translucent or luminescent, right? They glow. Now I can take that glow gene from a firefly and I can stick it into a plant, and that plant will also now glow. Okay, so we have glowing plants. We also have glowing kitties. So we can take that same firefly gene and put it into the cat's genome, and as it develops, all of its cells will also fluoresce, much like a firefly. You can now have there's now glowing kitties and dogs and monkeys and pigs, and there's no reason in the world why they couldn't make babies, human babies, to glow, but there's a thing called ethics that gets in the way. Okay? So, but it's very simple. It really is. When you go to Myers and you see in the fish store those neon green, orange, and yellow and purple fish, those aren't real, right? Those things aren't real. They are they're genetically modified, and they've done the same thing. They've taken that fluorescent gene and they've stuck it into the fish embryo, and they've made these glowing fish. Okay? Now some of them are real, but those big goldfish-looking things that are purple, no, those aren't real. So those are just genetically modified organisms. And you hear a lot about GMOs nowadays, right? There's a big push that we should know what in our diet is GMO, genetically modified organism. Most of the cornflakes you eat are genetically modified corn. Now, why was it genetically modified? So that we could grow bigger, taller, stronger, less corn plants that are more resistant to drought and more resistant to pests. So we can take a gene that gives wheat drought resistance that we discover and put that same gene into a corn plant. And now the corn is drought resistant. And we can take a gene we find for insect resistance and put it across other organisms. It's why we can now take the human insulin gene for making insulin and put it into bacteria. In E. coli, bacteria are now making human proteins for us to sell and give to diabetics. This is the whole basis of genetic engineering, taking one gene from one organism and putting it into another organism within an ethical framework, we hope. Right? Now, there's no evidence that I know of. There's a lot of fear, but there's no evidence that genetically modified organisms are going to hurt you. If you eat cornflakes made from a genetically modified corn plant, that mutation or that change in that corn plant is not going to somehow make you pest resistant, right? So this is not the way it works. So by consuming a genetically modified organism, you are not going to suddenly become a walking zombie or start glowing in the dark. So there's a lot of fear out there that is being propagated, but is not scientifically backed. So that's what's cool about the genetic code. It is redundant, it is ambiguous, or it is not ambiguous, and it is nearly universal. And a couple years ago, I asked a student, what does that mean? And they told me it's used in almost every country. It's not quite what I mean by universal, right? Universal means it's used in almost every organism on the planet, whatever that organism is. There's one more piece to this that will help you make sense of this, and that is the idea of reading frame. So the DNA has the code. The DNA is transcribed to mRNA. The mRNA then goes to the ribosome. The ribosome needs to know where to start. Right, so there has to be a place where the ribosome knows to start reading the protein, and there has to be a place where the ribosome needs to know to stop reading the protein. And that's the idea of a reading frame. So, I don't have an eraser, so I'll do it over on the other side. So, the idea is that I'm going to write a sentence. Oh, maybe this is, oh, that's an eraser. Okay. It's a newfangled one. Okay, so I'm going to write it on the screen, because I need my folks at home to hear this. So if I had a sentence, and the sentence is simply this. I had the big red dog. Okay. Now I'm purposely choosing small letters that are 3 letters long, right? So each 3 letters represents another codon, another triplet. The ribosome, just like when you're reading, you know to start reading a sentence because you see a capital letter. So too, you know to start. So the ribosome knows where to start, and also the ribosome knows where to end, sort of like a period in a sentence. You know, your eye looks for the capital letter and reads, and then it stops or pauses with that period. If, for example, the ribosome did not start with the, but instead started with H and went HEB, right? You see what's happening? So if the ribosome did not start with the T, but instead started with H, it went in 3 letters. It would go HEB, HEB, and then IGR, and then EDD, right? And what are we getting? Junk, right? So the ribosome needs to know where to start. So this idea of reading frame is also important in this world of translation. I mentioned tRNA. I just want to help a couple of students who I know struggle with this. So this is that lunar landing molecule. That's the tRNA molecule. This tRNA only has 1 amino acid attached to it, Lu. Now, those 3 letters down there are called what? Codon or anticodon? The anticodon. And the anticodon is going to specifically bind with these 3 letters called the codon. So the idea is that tRNA is going to transfer or bring in the correct amino acid. And how does it know which one to bring in? Those 3 letters, right? So very specific, very specific. So again, those words, anticodon, found on the tRNA, codon found on the RNA, on the mRNA. They find each other. Yep, they find each other. The ribosome made of 2 parts. Nothing fancy there. There's a big part and a small part. And the ribosome is so cool because it can, again, it reads and allows for the mRNA and the tRNA to come together. So in a way, the ribosome, I'm kind of stretching it, but the ribosome is kind of like an enzyme. Okay, in a way, because what is it doing? It's bringing together 2 pieces, isn't it? The ribosome is bringing together the mRNA and bringing together the tRNA and allowing them to interact, okay, to create the protein. So here's the punchline. If you have a point mutation, that is, you have a single change in your DNA. Now, that change could be a substitution. An A could now be a T. Or there can actually be mutations where there's additions or deletions. You can add a letter, take away a letter, add 2 letters, add in 2 letters. Those sorts of mutations, very small point mutations, can lead to dramatic differences in protein structure. So this is where I wanted us to go with this conversation. So on the top is a normal DNA sequence. Actually, it's RNA, isn't it, as used. So AUG, AAG. If you look on the table, AUG will give you MET, AAG will give you lyse, UUU will give you Phe. UAA is one of the 3 stop codons, so the protein stops being made by the ribosome. In this particular example, there's been a deletion, and this letter U has been lost. So there's a deletion here. So what does the ribosome do? It just starts at the beginning. AUG goes by 3, AAG. The next one is going to be UUG. Now, that's not what it's supposed to be. And as it continues in groups of 3, look what happens. It actually even loses the period, right? It loses the stop codon. It's like some of your sentences next week, these run-ons, they just go on and on and on, right? Just kidding. Really, not really. So it's that idea, right? It just keeps on going. And just so you know, I'm not grading you next week on like run-on sentence grammar kind of stuff. So just keep on writing, put your sentences together. What if instead there was an extra nucleotide put in like on the bottom? And AUG starts, but then all of a sudden, boom, UAA comes along. UAA is one of those stop codons. If this protein, whatever this protein is at the top, if this protein was necessary for the life of the cell, and now that mutation caused either a very short version or a garbage version of that protein to be made, that cell can't survive, right? That cell is going to die. That was the example where if a mutation occurs and the cell does apoptosis, dies, it's not always by choice, right? But the cell can no longer go on because of that mutation. Just another couple of examples. Sometimes these mutations can be groups of nucleotides. In this case, you've lost all 3. Now, in this case, again, you start off with MET, and then you lose an entire amino acid, and then you continue. You still stop in the right place. In my example, this would be, rather than the big red dog, right? This would be like the. And what got lost was the word big, right? And so, you know what? That may still make sense. You know, the red dog may still make sense. That protein may still be somewhat useful to the cell. It won't be as useful, but it may not be death to the cell because it lost that. And then a couple of other examples. This one is a unique situation where I've switched a U for a C. So in the normal, there was a C. In the mutation, there's a U. But look what happened. GGC and GGU are both going to code for gly. What is that demonstrating? Redundancy, right? In other words, there's 2 ways of saying gly. The DNA did have a mutation. There was definitely a change in the nucleotide. It got changed from a C to a U. But that mutation did not result in a change to the protein. We call that a silent mutation. It's definitely there. There's a change in the DNA, but it did not show up as any meaningful change. Silent. Silent mutation. And then there's just a couple more examples of the same thing for you to kind of look at and practice. So on Wednesday, I will give you a DNA sequence. From that, you'll need to figure out what the mRNA sequence is just by complementary C goes to G and A goes to U. And then you'll look at the table that I'll provide, and you'll figure out what the amino acids would be. That's what you need to be able to do from this. Here's the take-home punchline. This is an example of a rather common disease caused by a single-letter mutation. And this is sickle cell anemia. Sickle cell anemia is a disease caused by a change in the DNA sequence of hemoglobin. Hemoglobin is a protein that carries oxygen in your blood. Normal folks is a part of the hemoglobin gene, CTT. It's T's, right? It's DNA. That CTT would be transcribed to GAA. That GAA would be translated to glue. Go back, it's right. You can check it. What happens in the mutation of sickle cell anemia is that the CTT is now CAT. Seems simple enough.
How does that?
There's a mutation. Environmental problems, or you inherited it, right? Some replication error in the past. CAT now is transcribed to GUA. GUA is translated to BIOL. Big deal, right? One amino acid. But that one amino acid change makes the protein almost dysfunctional. So what happens, these individuals still live, but they live with some serious problems. On the top, you see normal red blood cells. And inside every red blood cell, there are millions of hemoglobin proteins. Those hemoglobins carry the oxygen. In sickle cell anemia, those hemoglobins cause a disshaping, a misshaping of the cell. Remember how squishy red blood cells are? You've seen how they can blow up and shrink. So you know they're kind of stretchy already. Those hemoglobin molecules start misbehaving, and they cause the cell to elongate into a sickled shape. Now, that sickled shape is now not able to travel through small blood vessels. Normal cells kind of bounce off blood vessels and travel through. These sickled cells get stuck, and they cause blood clots and blockages of blood flow. Now, that blood flow, if it gets blocked to a finger, it'll be painful, and that patient might lose their finger because the cells didn't get oxygen. They'll live, though. If, however, that blockage goes to their brain, their kidney, their spine, some other critical organ, it can be a life-threatening situation. The other thing that happens is that the hemoglobin no longer carries oxygen as well. So these patients are also anemic. So there's really two parts to the sickle cell. It changes the shape, and anemia, weakness. They don't carry oxygen well. So these patients are kind of sick. They're kind of weak. They have to be very careful on what they do. And during these sickling episodes, they can be life-threatening, and they find themselves in the hospital ER quite often. Question?
Why is it the tendency of people with this? They say the stereotype is African,
Yep.
but it's Mediterranean.
This disease is more common in people along the equator, Mediterranean and African. And it has been shown from an evolutionary standpoint that those with sickle cell, not with the full-blown disease, but those who carry it in their genome, are resistant to malaria. And where do you find malaria? Along the equator. So there might be a built-in reasoning for sickle cell anemia. Even though you have this problem, you have some resistance to malaria, which would kill you otherwise. So you've got sort of a trade-off, evolutionary trade-off. But you don't see sickle cell anemia anywhere near as much, if at all, in people from the poles. Okay, good connection. So again, just another example of one of these problems. So look at these problems, make sure you understand what's happening and how a change in the DNA or RNA can make an effect in the protein. That's really the take-home lesson. And to understand that, you had to understand a little bit about transcription and translation. That does finish up Chapter 3. It's been a long way to get it through Chapter 3. And look at this slide, just as a quick review of the organelles. So go through this, make sure that you can identify these organelles and know their function, right? As a review, make sure you can do that. So now it's time to move on to Chapter 4. I've got about 10 minutes, and that's all I need to get started on Chapter 4. Chapter 4 is going to be a discussion on histology. Okay? So moving on, Chapter 4, histology. Most of this will be a review. I will only have time right now to discuss epithelial tissues. We'll get on to connective tissues and a little bit about muscle and nervous tissue at the beginning of Monday. And then Monday, the rest of our time, we'll be looking at skin. And skin's a very easy chapter, very straightforward, Chapter 5. So one of the cool things to think about when you're going to bed and you're dreaming about anatomy and physiology, one of the new dreams you can have is, how is it that all of us, trillions of cells, came from one cell? Right? We were once all a zygote. The egg and sperm come together, form that one cell. That cell divides 2, 4, 8, 16, and eventually, here we are, trillions of cells later, with specialized cells making muscle and kidney and nervous and all those different kinds of cells in our body. Pretty cool story. So just to kind of give you the overview. So first, we started off as a zygote. Egg and sperm come together. That cell is going to split and split and split. And after about a week, that group of cells is going to be called a blastula. Now, the blastula is the same state that we saw in the mitosis slides, it was a fish blastula. It was a ball of cells actively and quickly growing. And that blastula is then going to begin to become 3 layers. Really cool, but the blastula will begin to form 3 layers of cells. And those 3 layers are now going to begin to dictate our overall body structure. The outer layer, the blue layer in this cartoon, is called the ectoderm, outer layer. The middle layer is the mesoderm, that's shown in the reddish pink, and the yellow is the endoderm within. There will also be a few cells called the germ cells. These are the cells that will go on to later in life become egg and sperm producing cells. But all of the cells in your body are coming from one of these 3 layers. That gastrula is somewhere around day 10, right, of embryological development. So within the first 10 days of your life, your embryo has already started making decisions. You cells, you're going to become muscle. And you cells, you're going to become kidney. And these cells, they're going to become respiratory system. What I want you to appreciate is that these, what kinds of tissues come from these layers. The ectoderm, the outer layer, will become your brain. Okay, so your brain is formed from the outer layer, as is your epidermis. And that makes sense. The outer skin layer, epidermis, will come from the ectoderm. From the inside layer, this one I think also makes good sense. This is going to be your tubes. This is going to be your digestive system, inside tube. This is going to be your respiratory system, right? Your throat and your windpipe, your trachea and your lungs. That's all going to come from the inside layer, the endoderm. Then the mesoderm, the middle layer, will become all of your connective tissues and your muscle tissues. Okay, so connective and muscle. I'm kind of simplifying that. But connective tissues and muscle tissue will come from the middle layer. Pretty cool stuff. I want you to understand stem cells just briefly. So this is always a controversy. So egg and sperm come together, that forms the zygote or the fertilized egg. That egg then divides, and makes that blastula. This is about the first 5 days of life. The cells in here are the cells that will become you and me, become the embryo itself. These are the cells that some scientists want as embryological stem cells, because these cells have not yet been assigned, right? They have not yet been told, will you become a muscle, a nerve, or a kidney cell? So these are the cells that they want. They want to take these cells, put them in a scientific environment, and from it now create other cells. And the idea is now we can take these cells and now inject them back into a patient and be therapeutic. This has not yet happened. Okay, it has not happened, and part of it is, yes, these cells can become any of these cell types, but notice this says cells. This is pancreatic cells. It doesn't say pancreas. This is liver cells. This does not say liver. This does not say a full muscle. So we've not yet been able to make full organs. We're getting closer, but not yet. Questions?
So is that why after you have a baby, they ask for like your
Yeah, the cord blood has in it stem cells. We don't know what to do with them yet, but someday maybe we will. So we'll freeze them and we'll charge you for it until then. Okay. Question? Yeah.
Isn't there a way that I did something in a biology class where you're not able to produce stem cells once the nervous system is ruptured, something like that?
No, stem cells are part of your body. Your nervous system does not have nerve cells. It does not have stem cells. So once you have spinal cord injury, it's not easy to recover or restore the nervous system. Okay.
Oh, no, it is. Once you destroy your nervous system, there's no way to alter the stem cells to produce.
Right, there are no stem cells. They will not reproduce nervous system. So if you damage your brain or spinal cord, there are no stem cells to come to the rescue. They're not going to repair. They're not going to replace. So what I wanted to tell you is that this is hugely controversial because you are destroying an embryo to get these cells. And this has not proven yet to be a helpful therapy. All of the therapy that you're hearing about in the U.S. dealing with stem cells is coming from adult stem cells. Basically, in your body, in my body, there are stem cells. I have stem cells in my skin that can regenerate skin. If I cut myself, right, I'm going to heal. Those are stem cells coming to the rescue. If I cut off part of my liver will regenerate in part because there are stem cells in my liver. So what we're finding now is that there are stem cells in our body that can now be used to treat disease. I have a friend who had a spinal cord injury from a wrestling accident in high school. He's now moving his limbs and potentially going to be walking. Now, it's still a ways to go, but there's some stem cell work that's going on. So there's a possibility of that, but all of that work has come from adult stem cells, not from embryological stem cells. So I really do think that in the next 10 years, this whole embryological stem cell debate will be behind us, because we will learn how to take stem cells from the actual patient. If a person's a diabetic, we'll be able to go into their own cells, take their own stem cells, and from it create cells that potentially could create insulin, and then put them back into the person of origin. So there would be no more issues with transplantation and things like that. So that's what's coming. Okay, that's what's coming in the next 20, 30 years, I hope. Isn't that weird?
Yeah,
that's cute. Okay, so let's go. Big picture. I got 5 minutes to get through some epithelial tissues. You know about epithelial tissues. You know, in fact, let's back up before that. There's 4 different groups of tissues, right? You know epithelial, connective, muscle, and nervous. What's new for you is that all of the connective and muscle tissues come from the mesoderm, that all of the nervous system comes from the ectoderm, the outer layer, and the epithelial tissues in your body actually come from all 3 layers. So the epidermis on the outside comes from the ectoderm. The mesothelium, that is your serous membranes, comes from the middle layer. Makes sense, right? Mesothelium, mesoderm. And the endothelium, the cells that line the inside of your blood vessels, come from the endoderm. So you'll hear different names for those epithelial layers in the body. So we know this. This is just review. Epithelial tissues line, coat, cover our body. They cover our glands. They line our cavities. And the majority of our glands are composed of epithelial tissues. You know that epithelial tissues are 1 or more layers of tightly packed cells. Because they are tightly packed cells, there's little to no space outside of the cells. That is, there's very little extracellular matrix, and there are no blood vessels that penetrate up through epithelial tissues. The cells are tightly held together, and these cells have polarity. This is new. That is, your epithelial cells know their top from their bottom. They're not just a blob. Okay? They know their top from the bottom. The top side is referred to as the apical surface of the cell. The bottom side is referred to as the basal, the basement. That's easy. The basal surface. And the cells are also tightly connected to one another through these intercellular junctions that I'll describe. So using that word apical, the apical surface, the top surface. So here on this simple epithelium, there's your apical surface, the top layer of the cells, and the basal surface, the bottom. The basal surface is the side of the cells that is glued down to the basement membrane. Okay. If you're looking at a stratified epithelium, the basal layer is attached to the basement membrane, the apical surface is at the free surface, and the cells are all attached tightly to each other by those intercellular connections. You also know that cells come in different shapes: squamous, cuboidal, and columnar. So this is all review. You know that the cells are, again, attached down to the basement membrane. What is down below the basement membrane? Connective tissue. And we'll be comparing and contrasting that a little bit. And remember that epithelial tissues do not have blood vessels traveling through them. That means that you can swallow a Dorito, right? And your esophagus is lined by those epithelial cells, and you're not going to bleed. It means that you can scratch your skin and you're not going to bleed. You don't have blood vessels right at the epithelial layer. It's down below. That also tells you, though, that the nutrients feeding those epithelial cells must be coming from underneath. So the blood flow is down in the dermis, down in the connective tissues, and those nutrients must diffuse. And we know a little bit about diffusion now. There are lots of nerve endings in your skin and in your epithelial layers. You can feel touch and temperature and pain. So clearly there are a lot of innervations, a fancy word for saying the nerve endings that are found in your tissues. And epithelial tissues are always regenerating. They're highly mitotic. They're replacing themselves all of the time. Okay? Replacing themselves all of the time. So we would say they're highly mitotic. So your skin cells are some of the fastest growing cells in your body. Those epithelial tissues are providing protection, think your skin, but they're also releasing things. I told you that many of your glands are epithelial. You are releasing hormones through your endocrine glands. You're releasing spit through exocrine glands, things that are going to exit your body like sweat. That would be an exocrine gland. And just like cell membranes provide a barrier for things in and out of a cell, epithelial layers provide a membrane, a permeability for things going in and out of organs and tissues. I already told you there are nerve endings there. And two things and we're done. Basement membrane. This is a little extra feature. The basement membrane is actually a double layer. I've not said it as a double layer in lab. So the double layer, the layer, there's two parts. Part of it's made by the epithelial cells, part of it's made by the connective tissue below. The part that's made by the epithelial cells is called the basal lamina. That lamina word, think laminated countertop layer. It's down by the basement membrane. The layer that's made by the connective tissues is the reticular lamina. You've seen that word reticular meaning a network. Together they make the basement membrane. So on this picture we see the epithelial cells on top, apical surface on the free surface, and then the connective tissue looks like wood grain down below. And you know that these cells come in three shapes, like I've told you, squamous, cuboidal, and columnar, and in multiple layers. I'm out of time. So what I'll do is on Monday, we'll pick up with these tight junctions. I'll then introduce to you connective tissues that you already know, really quick. I'll show you what muscle looks like under the microscope. Do go ahead and do Chapter 3 homework. You've got it all. Chapter 4 homework, start it, because most of it's histology, and you've already had histology in lab. So you shouldn't have much trouble doing Chapter 4 homework now. And then just save Chapter 5 homework for next Monday after we talk about skin.